Welcome to the pharmaphorum podcast. This is web editor Nicole Raleigh, and today I'll be speaking with Kelly Gold, who is CFO of CAMP4 Therapeutics, a biotechnology company advancing a new class of antisense oligonucleotides, or ASOs, medicines designed to selectively restore natural protein production in diseases caused by gene underexpression. Welcome, Kelly.
Thank you so much for having me, Nicole.
So today, Kelly and I will be discussing gene underexpression and specifically how SYNGAP1 related disorder negatively impacts synaptic signaling during critical periods of brain development, leading to a range of cognitive, epileptic, behavioural and motor symptoms. But before we sink our teeth into that, Kelly, I was wondering if you could briefly introduce yourself for our listeners and describe the journey that led you to doing what you do today.
Sure, thank you. I am the Chief Financial Officer at CAMP4 Therapeutics, and I've been with the company for 9 years now. My career has always been in healthcare in one form or another. Prior to CAMP4, I was in the finance and rare disease unit at Biogen, working on the hematological programs that were ultimately spun out. Also, I was part of that group during the launch of Spinraza for spinal muscular atrophy, which was a really important medicine about a decade ago for a similarly very devastating disease. Prior to that, I worked in investment banking in the healthcare group at Deutsche Bank, and I started my career as a mechanical engineer designing BSL-4 laboratories for infectious disease research. So always in one form or another, touching healthcare and human health.
Absolutely, a thread throughout. So let's turn to that healthcare side of things. I was wondering if you could tell us more about gene underexpression generally. I believe it is the underlying cause of more than 1,000 diseases. Is that correct?
That's correct. So we think of diseases caused by underexpression of protein as being haploinsufficient diseases. So these are diseases where the disease state is caused by having one healthy allele producing a wild type amount of mRNA, which is ultimately translated into healthy protein. And then your second allele is mutated in some form. It's either not producing anything at all, or it's producing something that is non-productive, is ultimately not resulting in a production of protein. And so at CAMP4, we have developed a platform by which we are engaging a target space called regulatory RNAs. These are non-coding RNAs that are actively transcribed during transcription within the cell's nucleus. And they are the master controllers of gene expression. And so what we found is that if we can engage them with antisense oligonucleotides, we can either tune up or tune down the expression of any gene. We, of course, are more interested in tuning up gene expression in order to address this 1,000 or so diseases that are haploinsufficiencies. And so what our platform is doing is actually turning up the healthy allele. So we are asking it to produce more than 50% of what it would produce in a healthy individual. And so by doing that, we can return protein production close to wild type levels. And what we've actually found in many of the disease areas that we work in is that we don't even need to restore protein production back to healthy levels. Even just increasing it up to within 70, 75, 80% of healthy levels is frequently enough to show a clinical benefit.
That's fascinating. So when it comes to the unmet needs in SYNGAP1 related disorder, what can you tell us about that?
SYNGAP1 related disorders is one disease in a larger group that are developmental epileptic encephalopathies. And so these are genetic epilepsies, but it's always very important to understand that within this group of diseases, the seizure burden is not the only burden of the disease. These are not simply seizure disorders. They have a neurodevelopmental component. They are always accompanied by intellectual disability. In the case of SYNGAP1 related disorders in particular, many of these patients, depending on genotype, are not at all able to communicate or very limited in their ability to communicate. They are very sleep disturbed. Many of these patients will get only 1 or 2 hours of sleep a night. And as you can imagine, being the caregiver of a patient who sleeps that little is quite challenging. So not just for the patient, but for the family itself. And so it's really this host of symptoms that we are seeking to address with our approach to treating SYNGAP1 related disorders, which is upregulating SYNGAP expression. So SYNGAP is a protein that is underexpressed in this disease. This is a true haploinsufficient disease. So these patients have 50% of the normal levels of SYNGAP protein. And the role of SYNGAP protein in your neuronal state is to basically regulate synaptic firing, like sort of said at its simplest level. And so when you have too little SYNGAP protein, you actually end up with an overabundance of AMPA receptors at the postsynaptic level. And that results in tighter synapses and higher synaptic firing. So you've got a more kind of excited baseline neuronal state than a wild type individual would have. And so what we are doing is upregulating expression of the SYNGAP protein. We are normalizing the number of AMPA receptors on the postsynaptic membrane, and we are reducing the sort of high level of neuronal excitability that's seen in the disease state. And so we will be entering the clinic with this program later this year. And our hope is that we will be able to address this very devastating disease for the 20,000 or so patients worldwide that we believe can be impacted by our therapy.
Thank you, Kelly. So before we go further into CAMP4's CMP002 study, I just wanted to stay more general for now and think about the antisense oligonucleotide or ASO space more broadly. You've been talking to what CAMP4 is attempting to do for these patients. I was wondering if you could describe for us the options as stand within the field.
Yes. So for this particular disease, there are no disease-modifying therapies currently in development. There are other companies and some academic groups that are looking at this disease at the very early stages of drug discovery. There is a group that is looking at gene therapy. That is, something that we've seen be transformative in a few different disease areas, spinal muscular atrophy being one of them. That work is very early. We, of course, would like to see the best therapies move forward for this particular disease and for any rare genetic disease, but that is in its early stages. There are a couple of other approaches also in early discovery for treatment of this disease. It's same modality as CAMP4, the antisense oligonucleotide, different mechanism of action, all in some form or another looking at restoring the missing SYNGAP protein. It's just sort of the underlying biological approach as to how that's being achieved that is different from CAMP4.
Thank you. So now to CAMP4 and the CMP002 study. I was wondering if you could share with us the details and maybe some of the data from that.
Sure. So we are very, very pleased to be the first company that will be launching a clinical study for the treatment of SYNGAP1 related disorders. We anticipate launching that study later this year. We have not released too many details of the study right now. I will share that we're moving directly into patients. In traditional drug discovery, phase 1 studies are frequently in healthy volunteers. In this particular disease area, because the unmet need is so high, we will be moving directly into patients. And so that is very important. We plan to be dosing in pediatric patients. This is a disease that typically will start showing symptoms around age 2, 3, 4. The time to diagnosis can vary, of course, depending on what diagnostic tools are accessible to each patient. But we would like to be able to get into patients as young as possible. So we are actively dialoguing with regulators on those particular topics right now. This will be a global study. So we plan to have sites in Australia, Europe, the UK, and are exploring other geographies in addition to those. But those are the areas that we will be initially launching the study in. And we plan to have at least 3 cohorts in this study. So it's certainly something that we plan to be flexible about as we see how the study enrolls and start to get some internal looks at some initial cuts of data. But at this moment in time, not having launched the study yet, our intention is to do 3 cohorts. In terms of the endpoints of the study, we are going to be looking at a number of different measures. As I said earlier, this is a disease that has a number of different symptoms, and we would like to be able to explore all of them. So we will be looking at our therapy's impact on seizure burden, its impact on sleep. We're also really interested in looking at markers of longer-term impact, including cognition, improvements in communication, motor function. And so we have a number of exploratory endpoints that we will be incorporating into this study because we have quite a bit to learn about how this disease will respond to our therapy. And so we were trying to collect as much data as possible in this initial study in order to help set up for a potential registrational study later on.
Thank you, Kelly. So with everything we've been discussing today, there's a lot of potential for the future. So if we think about the sort of future potential for targeting regulatory RNA, and not just in association with explorations in SYNGAP1, but also outside of those bounds, where next, so to speak, for this field of study?
That's a great question. I think that SYNGAP1 related disorders very quickly rose to the top of the list of diseases that we were interested in because there is significant unmet need. There's actually a very nice mouse model that we have used to generate much of the preclinical data on which we have based our thesis around the program. That's always very important, particularly in rare disease. Not all rare diseases have a good animal model, and that's very important for small companies like ours that need to create some sort of proof of biological concept in an in vivo model system before investing the substantial capital to go into patient studies. And so we are quite pleased with the progress in SYNGAP1 related disorders. I should also note that there is a very sophisticated and well-organized patient advocacy group of parents, caregivers, allies to this particular patient population. And they are actually very instrumental in moving forward this program in the sense that they have invested quite a bit in sponsoring a natural history study, and that natural history study will be important as we contextualize our own clinical data in the coming years. They also have done a very nice job of establishing a registry and really sort of evangelizing the state of the developmental and discovery field for this disease. So not just CAMP4's therapy, but all therapies that are being looked at earlier in the competitive pipeline. And so that registry and the knowledge of where patients are and where KOLs are will help us to think about where to establish our trial sites to better enroll the study. In terms of where we go next beyond SYNGAP, there are a number of other opportunities that fall under that DEE umbrella I described, those developmental epileptic encephalopathies that phenotypically are very similar to SYNGAP. And so they will have a similar sort of disease burden. They are also haploinsufficiencies, so squarely in the strike zone, if you will, for the CAMP4 platform. That's really where we can best apply our technology. And so there are a number of those that we are already working on. We've not publicly disclosed the names of those targets and diseases yet. That will change in the coming months. We will start talking more openly about the other ones that we're looking at. But I think that, certainly once you do one discovery and development program in this disease area, there are many learnings that can be applied to similar diseases from a regulatory endpoint design, interacting with the patient group. All of those things are things that will translate nicely to the earlier pipeline. But CAMP4 is very focused on the CNS in general. We are not a rare disease company. We are currently focusing a pipeline of rare disease programs, but the platform in general is broadly applicable across all therapeutic areas. We have a strong interest in the CNS in particular because there are a number of haploinsufficiencies. It's really a good area for us to address the modality that we use. You can deliver naked oligos, so that's an unconjugated oligonucleotide intrathecally. And we've been able to show in the SYNGAP1 program that with intrathecal dosing, we are able to get very nice distribution throughout the brain regions that are relevant to the disease state. And we believe that's also transferable to similar diseases. We are very excited about what comes next. We're doing quite a bit on our own as a relatively small public company. We also look to partners to help us to capitalize on the potential of our platform. We have one such partnership with GSK that we announced late in 2025, and we are actively discussing other partnerships that will allow us to fully realize the potential of our platform in the CNS and beyond in order to bring as many therapies as possible to patients.
Thank you, Kelly. I mean, it's hopeful indeed and necessary indeed. So to my final question, given everything we've been discussing in terms of CAMP4's platform and the unmet needs within this field, what would be your closing thoughts for our listeners today?
I think there is a lot of opportunity right now, not just using the oligo modality, but really in RNA targeting medicines in general. I think we've seen a number of successes in this field already. I think targeting RNA directly is an excellent way to go straight to the source. When we think about disease modifying therapies, historically, many of them have been replacing something that is lost by introducing it externally. I think what we really love about our approach in particular is we are working with a machinery that is already there in the nucleus of every cell. And we are just figuring out how to make it work for us in order to improve the disease state. So we like to say we are addressing the disease right at its foundational levels. And I think there is a lot of broad applicability from CAMP4's platform as well as other RNA targeting platforms and companies in the field. And I think that this is really going to be the next area of evolution in terms of introducing a completely new target space for our industry.
Thank you for your time today, Kelly. It's been a pleasure.
Thank you so much.
And that concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous installments at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on Apple Podcasts, Spotify, Overcast, Pocket Casts, Podbean, and pretty much wherever else you download your other podcasts from. Just search for pharmaphorum and follow and hit subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us on Blue Sky and LinkedIn at pharmaphorum. That's all for now. Thank you for listening.